pleasure to talk to you about some of the work we've been doing. Large part of this work that I'll talk about today has been funded by the National Pork Board. Some of it's been funded by the Iowa Pork Producers Association, and then also some of it's been funded by the USDA, National Institute for Food and Agriculture. So there's a group of us at Iowa State, quite a few more than just the three of us, and I'll get to some acknowledgments at the end, but the three of us have been looking at seasonal infertility and the role that heat stress plays in seasonal infertility the last few years in collaboration with Lance Baumgard, myself, and Aileen Keating. And so one thing that most people in this room are probably fairly familiar with is the incidence of seasonal infertility, and this is this predictable dip that we experience in the industry almost every year. Every once in a while you'll have a year, 1 out of 10, that maybe it doesn't show up as severely as you would see it. But in many commercial systems, it's a very predictable dip. About weeks 28 to 32 of the calendar year, or the trough of that dip, and then about 4 weeks on either side of that will be a period of time where there's compromised reproductive ability. So those would be the services that occur July and August and the beginning of September. Those are marked or result in reduced farrowing rates as well as reduced behavioral estrus demonstration during that period of breeding. The most susceptible group in that period of time are the parity 1 weaned sows. So after those sows that are weaning their first parity, which obviously also makes up, from a parity distribution, the largest group of the sow herd as well. So there's a lot of different causes or proposed causes of seasonal infertility. The one that we're investigating in our current projects was heat stress, trying to understand what role heat stress has and what contribution it makes or how it contributes to seasonal infertility. Obviously, there's the seasonality issue in and of itself. Photoperiod has also been proposed to have an effect on reproduction as well as it does in many other species such as sheep and horses. And then there's also the combination of those factors or other things that we're not fully aware of. One of the first projects I did when we got to Iowa State was we developed an in vitro heat stress model to really look at the direct effects of heat on a pig's oocyte as it undergoes maturation. And so I won't get into all the details here, but what we did was we heat stressed while the oocyte was maturing in vitro. We subjected it to heat stress at different time points. And so what this modeled in a pig is really what's happening as a sow's coming into estrus and ovulates. That's what's happening during this 42 hours and during heat stress. So that time period preceding ovulation and then during and behavioral estrus. And so one of the things that we discovered in that initial project is that heat stress compromised the ability of the oocytes to mature. So these two shorter bars here represent those oocytes that were subjected to heat stress during the early stages of maturation. So in those oocytes that were subjected to heat stress later in the maturation process were not as inhibited as much as the earlier phase of heat stress or the earlier stage of heat stress exposure. So not only did it compromise their ability to mature normally, but it also, we took those and did in vitro fertilization with them and looked at their ability to develop to the 4-cell stage. And that's kind of a developmental threshold during pig embryonic development. And we didn't see an effect of whether or not those oocytes, if they were heat stressed, we didn't see a suppressed ability to develop to the 4-cell stage of development. But what we did see is their ability to develop to a blastocyst. So we had this heat stress exposure happening during maturation of the oocyte, was not immediately evident in the early embryo development, but had dramatic effects on the development after the 4-cell stage of activation of the embryonic genome that occurs at the 4-cell stage in the pig. So this is really what got us thinking to put in a proposal to look at heat stress during the follicular phase, because we thought that the oocyte is particularly sensitive to heat stress during seasonal infertility. And so the reason that we thought that, here's a little more background from some different things that we've been looking at in our groups. Heat stress could have a direct effect on the oocyte, as we indicated with our in vitro study, but it could also have an indirect. So a couple of the things that happen during heat stress in pigs is that you have an increased amount of circulating insulin. So despite the fact that pigs go off feed and reduce feed intake during a heat stress bout, they actually have increased level or increased production of insulin. So that's counterintuitive why that would happen, but it happens nonetheless. And we also have looked at the ovaries and find that the receptor for insulin is also upregulated in the oocyte itself. So you have this increased insulin signaling that happens to the ovary and multiple things downstream of that, including activation of different enzymes and factors in the ovary and oocyte that can compromise the quality of that oocyte and its ability to produce an embryo. We've also shown in different studies that during heat stress in pigs, one of the things that happens is there's a vasodilation and a redistribution of blood flow to the peripheral, to the surface of the skin, because pigs can't sweat. The redistribution of the blood flow to the surface of the body is a mechanism or an effort to reduce body heat. So for the animal to maintain its blood pressure, if you have vasodilation at the surface, you have to have vasoconstriction in other areas. And vasoconstriction in the enteric system or in the gut causes a phenotype known as leaky gut, where the barrier of the gut system starts to break down, and that allows endotoxins such as LPS and other bacterial endotoxins to get into circulation. And so one of the pathways that endotoxins, when they're in the body, one of the receptors that they bind to is called toll-like receptor 4. That's also expressed in the follicle and in the oocyte. So these were two other indirect mechanisms that we thought heat stress could cause, based on the phenotype that it induces in a pig, could compromise reproduction. So the study that was funded by the National Pork Board, one of the things that we wanted to identify is during the wean to estrus interval in a parity 1 sow, at a farm in a commercial setting in a large system that experiences seasonal infertility annually, what is the role of heat stress during the wean to estrus interval in parity 1 weaned sows? And so we wanted to look at AI service rate, farrowing rate, litter size, and then compare all those variables, those reproductive variables, back to the indicators of physiological indicators of heat stress during the wean to estrus interval in those sows. And so the hypothesis was that the sows with the worst reproductive efficiency during seasonal infertility would be associated with those sows that were most susceptible to heat stress during the wean to estrus interval. So we did this in the summer of 2013 and spring of 2014, where we collected data on 450 parity 1 weaned sows over 4 weeks in the summer, and then we did 420 or 419 in the spring when reproductive performance was at a peak in this system. So what we did during the wean to estrus interval was we measured rectal temperature 5 times a day, skin temperature, respiratory rate, and we did that for the 7 days post-weaning 5 times a day. And we also collected blood samples 24 hours after weaning and again at 72 hours after weaning. And so we did 4 weeks in each of those time periods to get about 420 and 450 gilts in each of those studies. So the litter data that we collected then was number born alive, number weaned, the wean date, service result, total and live born on the second parity. And we really wanted to then retrospectively analyze, are the ones that were the highest rectal temperature, highest respiration rate, is there a correlation with reproductive performance during seasonal infertility? And sadly, our like many good hypotheses, were found to not be supported. And so what we saw here was that there are some correlations, but a lack of real correlation demonstrating that the sows with the worst reproductive performance was a result of heat stress. And one of the things when we went into that farm that we realized that was different than when we planned was that all the gilts are moved into, the parity 1 wean gilts are moved in front of the cool cells for the wean to estrus interval. And so they're actually, it was actually in hindsight ended up being quite a good study because we were able to show that you still have seasonal infertility despite the lack of heat stress. So what you see here is just with wean to estrus interval, the p-values in some of these cases are pretty low, but when you look at the correlation coefficient, it's really explaining very little of the variation in reproduction. Rectal temperature, skin temperature, respiration rate, all three of those really did not have a strong relationship with reproductive performance in any of those analyses that we did for either season. So this is the rectal temperature based on season during the wean to estrus interval. We didn't see a difference So that would be that'd be kind of the cardinal indication of a heat-stressed animal, would be an elevated rectal temperature. The other indication of physiological level of heat stress would be an increased respiration rate. So an animal could mitigate heat by increasing respiration rate without changing core body temperature. But we actually saw the opposite of what we would expect, and that respiration rate was actually slightly lower in the summer. And the reason for that is, or at least our hypothesis for that, is that the cool cells are running pretty effectively in the summertime, but weeks 12 to 15, the cool cells aren't up and running yet. And so we actually saw a slightly higher, although not different, respiration rate in the springtime. So again, not supporting the idea that the summer seasonal infertility dip was associated with an increased heat stress response. Skin temperature, we also recorded that, and similarly, as a result of, I think, the location of the cool cells and the location of the parity 1 wean sows in relation to the cool cells, we had a lower skin temperature in the summertime when the cool cells are running as opposed to the springtime, and that makes sense too. So if we look at the reproductive performance, this is where you see really the consequence of seasonal infertility on the industry and how much it really costs the swine industry. If we look at the farrowing rate, so farrowing rate would be the number of litters produced divided by the services that were made. It's still around 89% in the springtime, although there's about a 10% reduction in the summertime. So significantly reduced farrowing rate during the summertime in this system, and that's consistent with the 10 previous years in that system. But we really didn't see a difference on litter born or total born alive, or stillbirths or mummified fetuses as well. So we did see also, along with the reduced farrowing rate, one of the other observations we made was the increase in wean to estrus interval. So of all those that were weaned, we saw about a 2.5 day increase in the wean to estrus interval of those parity 1 wean sows. So if you look at the data, breaking it out a little bit more, evaluating the wean to estrus interval during seasonal infertility and farrowing rate by season, there's two areas that where reproduction is really compromised during seasonal infertility in this system. And one is the number of parity 1 wean gilts that are serviced. So what we see is a little more than a 10% reduction in the ones that were serviced. So this reduction isn't included in the reduced farrowing rate. So there's a 10% reduction in the number of gilts that are weaning their first litter that demonstrate behavioral estrus during weeks 28 to 32 of the calendar year compared to weeks 12 to 15 of the calendar year. Of those that are bred, the farrowing rate in the springtime is 91% and 82% in the summertime. So not only do you have a 10% reduction in demonstration of behavioral estrus, but you have another 10% approximately reduction in farrowing rate. And that's where you start really adding up piglets, is when you're having your gilts not come into heat, and then the ones that are getting serviced aren't producing piglets. Some of that can be recovered, right? So of the gilts that come into estrus and are serviced within 7 days, there's 88% right in the springtime. Their wean to estrus interval is around 4 days, and it's about 4 days in the summertime as well. What you see, though, is that the ones that are serviced after 7 days on average are about 24, 26 days, depending on the season. It's just that the percentage of those is much smaller in the springtime than it is in the summertime. So if you were to calculate, based on the reduced number of sows that are coming into heat, parity 1 sows are coming into heat, and the reduced farrowing rate, and you start adding that up, for every 100 sows on this project, that costs us about 224 piglets that aren't produced as a result of that reduction in the number that showed behavioral estrus and the reduction in the farrowing rate. So if you continue to, now that's the loss if you just weaned, or if you culled any sow that didn't breed after 7 days. But if you keep those sows around and you get an average service date of about 24 to 25 days, you can get some of those pigs back. You can get about 140 pigs produced out of those sows that have that delayed onset of estrus. But you're still losing. Sorry, you can recover some of those, but you're still losing 140 piglets as a result of the compromised reproduction in that season for every 100 sows. So that's 1.4 piglets per parity 1 wean sow during the seasonal dip. And you start multiplying that by the size of our industry and the percentage of parity 1 sows in the industry. It's a huge number of piglets that we're not producing. So as I mentioned, one of the indirect mechanisms that we thought would maybe be compromising fertility is associated with that increased insulin and increased endotoxin circulation. So we measured insulin in the blood on day 1 and day 3 following weaning, and as well we measured a compound called LPS binding protein that would be an indicator of how much endotoxin is circulating in those pigs. And we did a retrospective approach on that. So we took gilts that farrowed, came into estrus within 7 days and produced a litter, and then those that came into estrus and did not produce a litter, and then also those that had an extended wean to estrus interval. And unfortunately, what we saw is that there was no difference in any of those in circulating insulin, and there was no difference in circulating LPB. So what we ended up confirming was that seasonal infertility is really bad, and it was kind of like we didn't figure out what the silver bullet was, but we figured out what a few of them weren't. And so we do know that the significant differences or losses in reproductive performance are a result of compromised wean to estrus interval, compromised ability to show a behavioral estrus within 7 days, and then the compromised farrowing rate of those that are bred. And we didn't see a difference in really physiological markers of heat stress or differences in endocrine markers that are typically associated with severe heat stress in other studies that we've done. So that was in a commercial study. And then the other thing that we wanted to do, because we were really trying to parse out what is the effect of heat stress during the follicle's development and during the oocyte's development. And so the other part of the project, at the same time when we were doing that project, we were doing a project at Iowa State University where we used about 250 gilts, and we wanted to determine if their heat stress tolerance early in life, if we could see what the variation in pigs are, or in developing gilts, what's their variation in the response to heat stress, and is that predictive of their reproductive success later during a heat stress bout. And so what the hypothesis that we were testing was that gilts that are susceptible to heat stress are more likely to have a reduced reproductive success later in life. And I think one thing that you'll see is that we're kind of changing how we refer to tolerance and susceptibility to heat stress after we did this study. And so this part of the project was done in 2 phases. The first phase, we took about 235 gilts. We had to do this in 5 replications because of the facilities that we were working in, that we could use about 48 animals at a time. And we brought them in, we acclimated them, we went through a thermal neutral period of 24 hours and the heat stress period of 24 hours, and we recorded daily feed intake and body weight. And one of the things then during this 24-hour period is we took 16 different time points, we took rectal temperatures on these animals. And then also another 16 times we took rectal temperatures during the heat stress period where we had a pretty robust 24-hour heat stress. And really what we were trying to identify is what is the response, what is the variation in a group of gilts that are subjected to heat stress. And so this is kind of what the data looks like. This would be all of them averaged out. If you were to look at this was the time period here about midnight to 4 a.m. when the graduate students went home and got a nap. So we quit collecting data during that peak time. But the blue line is the average of those 240 gilts during the thermoneutral 24-hour period. And the red line is how the average of their response. Okay, so for each individual then, we wanted to calculate the difference. What's the change in their response? And so that was the average from this time point to this time point and during the heat stress divided by their 24-hour thermoneutral time point or response. And so then we did some analysis and we looked at the rectal temperature during heat stress does not explain the decrease in feed intake. So one of the common thoughts on heat stress is that the reduction in feed intake is the animal's attempt to mitigate the effects of heat. And that's logical and that's a legitimate response. But what we did find is that the severity of heat stress does not explain the amount of feed intake reduction. So while almost all of them went off feed, there was a couple that continued to eat, but 98% of them reduced feed intake during the heat stress response. But that was not correlated with how hot those pigs got from a heat stress standpoint. So what I'm saying is that some animals get really hot, but they continue to eat and lose less weight during a heat stress bout. So the heat stress feed intake also was not correlated with the heat stress rectal temperature. So that's what you see here, kind of plotted. That as an animal's rectal temperature goes up in a heat stress environment, it is not predictive of how much feed they'll eat in that environment. And so we also looked at their body weight change during heat stress, and we found that the severity of heat stress also did not impact the amount of body weight that animal lost during that heat stress bout. So, and then we also saw that the change in body weight is not completely explained by the reduction during heat stress feed intake. So there's a couple different things there that are happening, and what we really came around to think about was that what we were originally going to call a tolerant pig, based on how hot they got, maybe isn't a production tolerant pig. And so a production tolerant pig would be one that would continue to produce in a hot environment. Those also may be the ones that get really hot but don't die. So when we first plotted this, what we did was we looked at how hot they got, the heat stress rectal temperature over here on the left, and the change in rectal temperature on the bottom. And you can see there's a pretty strong correlation that the lower the rectal temperature during heat stress, the less the change was, so that made sense. And so we looked at these extremes here, and we wanted to try to find 45 to 50 gilts that were down here in this low end that we were originally considering to be tolerant of heat stress, and another 50 that were up here that we would consider to be susceptible. And so one of the things, when you look at that, one of the things that we identified is that retrospectively, those that got the hottest during heat stress were hotter already to begin with in thermal neutral conditions. And then that was exacerbated when you put them in a heat stress condition. So they already have a higher resting or biologically higher set point, thermoregulatory set point in a thermal neutral environment. So we use those gilts from the extreme ends, and we conducted boar exposure beginning around 160 days of age. Found those that were cycling and had at least a normal cycle before 220 days of age. And we utilized 48 of each of those groups for the second phase of the project, which involved moving into another facility where we could heat stress all 100 of them, or all 96 of them at once. And so during the acclimation phase, we supplemented them with matrix for 14 days because we wanted to make sure that the heat stress exposure on those gilts was occurring when they're all in the follicular phase of the estrus cycle. So they're all 5-6 days coming into behavioral estrus. And so at the end of matrix, the last day of matrix was also the first day that we initiated this cyclical heat stress conditions where we ramped them up to 33 degrees for 12 hours a day. And we had to let them cool off through the night, primarily as a result of doing the boar exposure and heat detection. And if they're extremely heat stressed, we weren't going to be able to find the ones that were in behavioral estrus, even though it was hot. So we had to cool it down to be able to identify gilts that were in behavioral estrus. So then for that next 9 days, they underwent heat stress, heat detection, behavioral estrus detection, and then insemination each day that they were in behavioral estrus. And after insemination, then have days 43 to 48 of gestation. We took them up to a harvest facility, collected the reproductive tracts from every one of them, measured the number of CLs, the number of fetuses, the crown rump length, as much of the reproductive parameters as we could on each of those animals. So they were all fed the same amount of feed during that entire phase 2. And again, we collected the fetuses, total uterine tract weight, ovarian weight, corporal lutea, etc. So one of the things, again, that we saw is this dramatic variation in thermoregulation. So this is the 238 gilts plotted individually. So you can see the amount of variation from some that are just pretty cool all the time in the thermal neutral conditions and some that are fairly hot. And during heat stress, you can see that variation increases pretty dramatically. And those are the ones that we selected as susceptible or tolerant down here and susceptible up here. And then this is kind of the chart of their heat stress response based on rectal temperature during that second heat stress that was 4 to 5 months later in life. And so even though it was 4 to 5 months later in life, the ones that we had classified as tolerant based on their ability to maintain a low rectal temperature during heat stress and those that were classified as susceptible still maintain that same phenotype. So the thermoregulatory set point, while it's highly variable across gilts, it's fairly fixed, meaning that it doesn't change over time. So what was interesting is that we had a very, very high pregnancy rate. These gilts were very hot during that 9-day period. I should also mention that we did this project, the breeding and gestation occurred between March 1st and March 9th, so it was during seasonally when reproduction is typically pretty good for pigs in Iowa and the Midwest. But what we didn't see was a relationship between fetal count and how hot those gilts got during that 9-day period following the matrix withdrawal. Where we did see a difference, and I don't really understand the biology of this quite yet. I have some ideas of why this happened, but nothing to confirm it, is that those gilts that had the higher rectal temperature at breeding had slightly heavier fetuses and had they also had a slightly longer crown rump length. So there's a little bit more of a developmental advantage in those gilts, or in those fetuses that were in gilts that were hotter during the wean to estrus period. So this is just kind of the same data showing the correlation there, that the fetal size was correlated with the heat stress rectal temperature response during estrus and breeding. So we did not see an effect on the number of corpora lutea. So we thought maybe compromised follicle development during that 9-day period would compromise the number of corpora lutea, those ovulatory sites on the ovary. We did not see an effect there. And we also didn't see an effect on the diameter of the CL based on how hot gilts got during the matrix withdrawal phase and follicular phase. And then this is just a chart of, one nice thing about pig reproduction is that you can count the number of fetuses and you can count the number of CLs. And because fertilization rate is so high in pigs, you can really get a pretty good approximation of embryo survival by counting the number of fetuses and dividing by the number of CLs. And one thing that we found is that regardless of how hot those gilts got, there wasn't a relationship with embryo survival. And so that was kind of, again, another one of those hypotheses where we weren't able to directly demonstrate that heat stress during the spring, during March, did not compromise the follicle or the oocyte's ability to become a viable fetus at day 43 to 45. And so one of the interesting things, though, that we did discover by doing this project is this relationship between the prepubertal rectal temperature in thermoneutral conditions is still correlated with their thermoneutral rectal temperature and later in life, post-puberty. And not only is it highly correlated, but it's also the prepubertal heat stress response is also correlated with the postpubertal heat stress response. But interestingly, the prepubertal thermoneutral rectal temperature is also very strongly correlated with the postpubertal heat stress response, meaning those gilts that had a lower resting thermoneutral rectal temperature had a lower heat stress response with respect to rectal temperature later in life. So 5 months later, it had predictive value. And so one of the things that's nice about this, we were able to leverage this project from the National Pork Board, and we had collected DNA samples on all 240 of those gilts. And we were able to secure some additional money working with Max Rothschild and Kwon-Suk Kim at Iowa State University. And we did a genome-wide association study. And so one of the things that I thought that I mentioned that we were kind of struck by is the lack of relationship between some of the production variables, such as feed intake and body weight loss, and the relationship with the thermoregulatory response to heat stress. And after doing some of the GWAS or genome-wide association studies, one of the things that we realize is that the phenotypes such as the rectal temperature during heat stress and respiration rate during heat stress are associated with different regions of the genome on different chromosomes than weight loss or feed intake during heat stress. So that made biological sense to us, or genetic sense to the biology of why we see a disconnect between the production efficiency response to heat stress and the thermoregulatory response to heat stress. And was the foundation of another project that we submitted to the USDA last year. So again, heat stress rectal temperature was not associated with feed intake or change in body weight during an acute heat stress. The heat stress rectal temperature did not explain decreased production efficiency. The one finding that we saw that was consistent was that fetal size was associated with elevated rectal temperature during heat stress during the estrus and breeding, and that the heat stress response based on rectal temperature following an exposure early in life was predictive of later in life, suggesting that while it's variable, it tends to be fixed in gilts. So some of that stuff is also one of the other things that we've looked at, because this was focused primarily on, up to this point, all this data that I've shown you has been focused on what's happening to the animal, the implications that has on the oocytes quality and the ability to produce a viable embryo in a pregnancy. One of the other areas that our group's been investigating, and I'm just going to kind of give the overview. We have done a lot of stuff in this area, so this is kind of the high points of it. But we've been looking at the effect of in utero heat stress and its impact on productivity. So there's a couple different models. I won't belabor this a lot. There's no clock in here, so I'm not even sure how I'm doing on time, but yeah, I'm about okay. So I won't belabor this a ton, but one of the things that we've looked at is the role of heat stress during early gestation and during gestation, what implications that has for piglets later in life. And so there's a lot of models that have looked at events that happen in utero and how that impacts postnatally that developing fetus when it becomes an adult. So some examples in different species are the intrauterine growth retardation models. So sheep are a really good model for that, where there's lifelong implications for compromised placental function early in life. Maternal stress is another one. So in rodent, there's been quite a few rodent studies where stressing out pregnant rats and evaluating those pups that were born from those rats have disrupted an abnormal ability to regulate cortisol production and glucocorticoid production later in life. There's very good examples of endocrine disruption during pregnancy. One example in humans is in the 30s, 40s, and 50s that women that were trying to that struggled with miscarriages, doctors were prescribing a drug called diethylstilbestrol, which is a very potent estrogen receptor agonist. And what they discovered after several years of doing that when daughters that were exposed in utero to diethylstilbestrol, when they reached puberty, they were developing cervical cancer at a really high rate. And so that's an example of where disrupting normal endocrine function during early embryonic development causes lifelong consequences, and actually even causes transgenerational consequences. And those girls, when they have daughters, also have increased probability of cervical cancer and reproductive cancers. So we evaluated or tested the hypothesis. We wanted to determine what the impact of heat stress on sows during gestation would have postnatally on those piglets. And so we collaborated at the University of Missouri on this project, and they have some nice environmental chambers that can facilitate the heat stress of sows and gilts. And so we did the initial study with 4 different treatments. We had gilts that were thermal neutral, both the first and second half of gestation, and then also heat stress the first and second half of gestation. And then we also had 2 groups that were kind of a switchover that swapped between thermal neutral the first half and heat stress the first half. We took those pigs, and I'm going to go through and just show a couple of the high points, but we took those pigs and brought them to Iowa State University, and we did several projects with that first replication of piglets. And one of the things that we looked at was at about 12 weeks of age, we took some ultrasound measurements, and we discovered that there was an effect of gestation on the size of the longissimus dorsi area at 12 weeks of age. And as we followed those animals, we looked at that again at 19 weeks of age. And again, we saw that on the back fat thickness, there was greater back fat thickness on those gilts that were exposed to heat stress early in life, or early in gestation. And so along with those lines, we also pulled blood at 19 weeks when we sacrificed those animals. And we saw that there was about a 50% increase in circulating insulin in those piglets that were exposed to heat stress, or that were in dams that were exposed to heat stress only during the first half of gestation. And so that was pretty intriguing. We used those animals and did some different analyses, some gene sequencing, mRNA sequencing, and protein analysis in those animals. But we also started at the University of Missouri a second replication of heat stress gilts to repeat this. And so on the second round, we used a different approach. We only did we only did thermoneutral the entire length of gestation, or heat stress the entire length of gestation to simplify the experiment. And then we did a serial slaughter experiment where we took pigs that were from those gestational environments, and we evaluated protein accretion and adipose accretion during the 30 to 60 kilogram phase, or the lean accretion phase, and again at the 60 to 90 kilogram phase of production, or the lipid accretion phase. And we also we postnatally heat stress them as well, or let them grow in thermoneutral conditions. But the emphasis that I'm going to share with you will be on the effects of the gestational environment, what happens to those pigs later in life. And so what you see here, this is during the lipid accretion phase, so during that 60 to 90 kilogram phase of production, what we saw was a pretty dramatic effect on the percentage of protein that was deposited in those pigs, and the protein deposition rate per day, and the fat to protein ratio. There was a very significant effect from the gestational environment on all three of those conditions. And what really that correlates to is a reduction in the body composition, or the amount of protein that's contributing to the body composition of those pigs. It's also resulting in a reduction of the protein deposition rate, or grams of protein produced per day. Conversely, we saw an increase in the body fat composition of those pigs produced from gestationally heat-stressed gilts, and we saw an increase in the adipose deposition rate. So collectively, we had an altered fat to protein ratio, and this was very similar, a completely different technique to evaluating what the effects are in these pigs compared to the first study, but very similar in that we saw an increased favor for fat to contribute to the carcass, as well as that elevated insulin in those piglets. So one of the things, I won't belabor, but we could go into a lot of details and talk about how epigenetic imprinting happens and how gestational events can result in postnatal phenotypes or observations in pigs. But one of the things that we did is we took what's a more molecular approach, and we took tissue samples from 96 different pigs that were subjected to different gestational environments, and looked at the genes and the proteins that are expressed in those tissues as a result of something that happened prenatally prior to in their first third or first half of their gestation. And just based on that, we saw that there were over 400 genes. So in each one of our cells, we have the capacity to express about 20 to 30,000 genes. Humans do. Pigs are about the same. And so what we found in liver, adipose tissue, and muscle was that anywhere from 400 to 1,000 genes were altered. The amount of expression was different based on what happened to them in their first half of gestation in those sows. And so I just had a student finish up some of this work, and he looked at, well, that's just the mRNA expression, the genes that are in that tissue. What about the proteins that are in that muscle? And so he's validated different proteins using a technique called Western blot analysis, in measuring specific proteins in those different tissues, and was able to demonstrate that there's a gestational effect on multiple proteins based on something that happened in the first half of gestation. Now has changed the amount of a specific protein, and in this case, heat shock protein 70, MEF2A. These are a couple different genes that are associated with that animal's ability to respond to heat stress, also their ability to produce muscle fibers. That was altered at 19 weeks of age after being born based on what happened to them in the first half of gestation. So I'll kind of summarize here what we've gone over. The heat stress has a direct negative effect on the oocyte. That's the in vitro study that we had looked at. Despite the lack of a physiological heat stress in the parity 1 sows, we saw a very substantial demonstration of seasonal infertility, mainly through the compromised farrowing rate, but also through that 10% reduction in behavioral estrus. And I have a few different ideas on mitigation strategies for that behavioral estrus, because some of those gilts that aren't coming into heat, we do have a reduced farrowing rate, but some of those gilts that aren't coming into heat are still fertile. We're just not detecting them in estrus to be able to breed them. So I think that's one of the things that we can work towards. That's probably some of the low-hanging fruit with regard to seasonal infertility, is figuring out how we can identify those parity 1 wean sows that are fertile but aren't demonstrating behavioral estrus. The thermoregulatory response to heat stress is really variable in gilts, but it's a fixed trait once that animal's born. What I didn't show you also is that that piglets produced from sows that are heat stressed during gestation, that those piglets have a higher thermoregulatory set point. So it increases their resting body core temp, or the core body temperature based on their environment that they were gestated in. And then prenatal heat exposure also compromises pork production through altering lean tissue accretion rate, as well as altering the amount of that body or of that carcass that's represented by lean tissue or by protein. And I shared some of that epigenetic or the maternal heat stress study with a group down in Australia back in November, and I went through it a little more detail than what we had time for today. And one of the comments I got from a very large producer, integrated producer there, was we have these fat carcasses that we see, and this seasonal increase in fat in the slaughter facilities. We could never figure it out, and then we were reading a couple of the papers you guys had published from your group, and it goes right back to the hottest period of gestation. That when those sows are in the first half of gestation, that's when you pencil it out. That's when we have the fattest hogs in the slaughter facility. So it is. It's a small effect, right? It's not something you watch close, but when you multiply that over 100 million feeder pigs, it starts to add up pretty substantially. So a lot of different people, as I mentioned at the beginning, contributed to this work. It's multiple groups. So the Aileen Keatings and Lance Baumgard group are two of the biggest collaborators in the seasonality and heat stress of reproduction at Iowa State. But there's also a large AFRI group that was funded by the USDA that contributed to this work. And then a lot of this work, the majority of the work shared today was funded by the Pork Checkoff and the National Pork Board, and then also some from the Iowa Pork Producers Association. So thanks for the opportunity to share, and if you have any questions, I'd like to do my best to answer them.
Got a couple minutes for questions. Wayne?
So on the parity ones, you showed summertime is really hard on those. But if they were maiden gifts and you bred them at the same season, would it be that dramatic?
I don't think so. We haven't done that study, but I don't think you would see it. I think that's one of the other things that we would like to do is to go back. So we really zoned in on that wean to estrus interval, and in hindsight, I think we missed the window where we should have been zoning in a little bit. So what we would like to do on a subsequent project is really zone in on what's happening during lactation and late gestation, and how does that correlate to reproductive performance later. And so one of the things that happens in mammals and pigs as well is that the oocyte that's ovulated during estrus actually was recruited from the follicular pool about 60 to 80 days before that. And so there's this window from recruitment to ovulation of about 80 days where there's opportunities to negatively influence the quality of that oocyte and take away from its developmental potential. And so the other time for that is when you go through, when a growing gilt's going through her first lactation, there's obviously a dramatic metabolic pull down on her during lactation. And you also have then a little warmer environment as well because of the heat mats in the summer. And then it's, the tolerance where you see a reduction in feed intake as a result of the environment in the room is pretty low tolerance. So, I mean, even 73, 74 degrees, you're going to start seeing some feed intake reductions in lactation. And so, if I were to go back and say where we want to go next with this, I would say to really narrow it down would be to look at the lactation room environment and then the interaction of feed intake by season during lactation. So I think, and I'm just kind of guessing here and sharing my ideas, but I think there would be a season by feed intake interaction. And you don't know that without going out and collecting data, but I think a gilt could tolerate reduced feed intake in the springtime that she can't tolerate in the summer.
So to assist the P1 in overcoming the deficiencies, what are your thoughts on administering vitamin E, or do you know any?
Yeah, I'm not real familiar with that. I think there's a lot of different thoughts out there right now on things that you can supplement during lactation to try to mitigate the effects of seasonal infertility. Some of them are different amino acids, different nutritional supplements during lactation. Vitamins would be another one. I think there's been some groups that have shown vitamin A effects on oocyte competency and the quality of a follicle. I think at NC State back in the late 80s and early 90s. So I think there's a lot of different things that probably need to be narrowed in on. The other thing I would say is that that we're starting to see is that genetically, based on this data and the GWAS experiment that we've done, that traits such as feed intake and average daily gain, it's really a different trait in the summertime than it is the rest of the year. And so I think a lot of times we consider it a single trait, but it's really a different trait in different environments.
Other questions for Dr. Ross?
Yeah.
You mentioned the farrowing rate, drops. What is the fallout mostly due to abortions, small abortions?
So yeah, that's a combination. I don't have my finger right on those numbers. So it's a combination of what some people call slip litter. Steve Pullman would call it a slip litter, right? But then there's also just an increased number of returns. So the number of gilts that just return 21 to 22 days. Then there's an increased number of preg check negatives at day 28. And then there's also an increase in the number that are day 28 preg check positive, but then end up not producing a litter. And so that's a pretty substantial component. I don't remember, based on these data, what that amount is, but there's a pretty substantial increase in the day 28 preg check positives that end up not producing a litter. And so, and usually by the time, in a lot of these, by the time you're getting a day 28 preg check, we're kind of through the rough season from an infertility standpoint. So retrospectively, I think there's still some programming that's happening in the follicle and in the oocyte, and it could also be on the paternal side too, which our groups haven't looked at at all, but that compromise placental function and placental development that ends up later in life not being able to, or later in gestation not being able to sustain a pregnancy.